Skeletons fueled the Cambrian Explosion. As the most important evolutionary innovation in the animal kingdom, skeletal structures have evolved multiple times across different animal lineages in various ways. It helped animals attain ever bigger sizes, while at the same time unlocking many additional abilities and advantages. Skeletons range from soft, hydrostatic systems to full body armor and everything in between. Vertebrates are renowned for their ability to build large and complicated bodies shaped from within by their intricate and highly adaptable internal skeletons. Yet there also lies a paradox in that skeletons both expand and constrain animal body plans. So how did skeletons develop in the ancient ancestors of animals in the first place? How was it even able to get started in the humble beginnings of a blob of cells suspended in the primeval seas? Will this be expected to be a universal trend for all life in the cosmos? And in order to evolve a life form rivaling human beings, is there really no way around an internal skeleton like we see in vertebrates? Let's try to find out. When it comes to the biomechanics of life, size matters, and it matters a whole lot. The cell membranes of tiny unicellular organisms are about as thin as that of a soap bubble, yet they rarely run the risk of being popped. And this has partly to do with the viscosity of water at these microscopic levels. For a protist, swimming through water is more akin to moving through molasses. It seems paradoxical that as creatures grow larger, they also become more vulnerable. But in reality, this is not really all that strange, because bigger bodies are subject to greater relative strains mechanically. You can build a small hut from sticks or clay, but not a skyscraper. Now it's important to keep into mind that cells are not merely water bubbles. They have a skeleton of their own called a cytoskeleton, and it consists of long molecular chains forming a network of filaments throughout the cell's internal cytoplasm. But when cells started to combine into ever larger bodies, this proved no longer sufficient. Multicellularity evolved in part because single cells began secreting specific compounds that allowed them to stick together, and this already provides some structural support. Cells of multicellular animals are often embedded in what is called an extracellular matrix. This is a web of polymeric chains of molecules that can directly bind to receptors sticking out from cell membranes, and in doing so form a so-called adhesome. This extracellular matrix was foundational for enabling early animals to grow larger bodies. And increasing body size seemed to have been a response to the rise of microbial predators about 800 million years ago that may be related to the sharply rising oxygen levels at that time. This is the Neoproterozoic oxygenation event and will be discussed in more detail in another video. One of the earliest animal branches, the porifera, better known as sponges, emerged at that time. Sponges create large structures with an external layer of flattened cells called pinacocytes and an internal layer of feeding cells called choanocytes, separated by a so-called mesohyl. The mesohyl is usually a mixture of collagen fibers trotted up by tiny skeletal elements called spicules that can consist of either calcium carbonate or silica. Sponges probably started out first with purely organic skeletons deposited as relatively thick fibers of collagen by specialized amoeba-like cells called sclerocytes. The next step was then to develop enzymes for encasing these fibers into silica crystals by using them as scaffolding and thus creating the characteristic spicules. During the Cryogenian period, when sponges first evolved, there was a lot of dissolved silica in the oceans due to increased rock weathering caused by the extensive global glaciations then. This abundance of silica may explain why sponges then learned to accumulate and then incorporate this material into their skeletons. Thus, sponges may have been the first animals to achieve biomineralization, which is the generation of hard, inorganic structures within or around the body. The next grade of animal evolution is represented by cnidarians such as jellyfish and sea anemones. These animals possess true tissues, meaning that a layer of cells is typically embedded on what is called a basement membrane. This kind of cell layer is called an epithelium and has the advantage of providing additional firmness by keeping any liquid or gelatinous masses it contains under pressure. Many groups of cnidarians also developed harder skeletons of varying types, both external and internal. This is an ancient group, so it makes sense that they would have tried many different ways of strengthening and protecting their bodies. However, their body plans are still quite simple, typically consisting of two primary tissue layers, the epidermis and gastrodermis, with a non-cellular mesoglea in between. It's the development of a third tissue layer, the mesoderm, that really started opening up possibilities. Triploblastic animals form internal body cavities called coeloms that in their simplest form are fluid-filled sacs acting like water balloons to inflate the body from within. The body shape can then be manipulated by systems of ring and longitudinal muscles. Annelids like rainworms, for example, have segmented coeloms that help them taper and thicken themselves in order to squeeze into soil and move themselves through or over many substrates. Another group of worm-like animals that use this technique for burrowing and foraging are the priapulids. Now, these creatures go all the way back to the Cambrian Explosion, and it is believed they represent the ancestral state of the ecdysozoans, a group that also includes arthropods. Ecdysozoans are characterized by having a tough outer cuticle that is secreted by the epidermal cells and typically a complex layered mixture of various organic polymers like chitin. Just like the keratin of your hair and nails, it is an inert, non-living material that cannot grow and therefore needs to be shed regularly. Still, the cuticle is normally flexible in most of the ecdysozoan branches, yet also in this state it does already constitute an exoskeleton to some degree. Only in arthropods has the cuticle become fully sclerotized, that is, hardened to form stiff plates and cylinders encompassing the entire body and all its extremities. This evolutionary process of so-called arthrodization of the entire body appears to have occurred in stages, one body region at a time. I will discuss how exactly this went about in another video. But what is interesting for our current focus right now is how the hardening or stiffening was actually achieved. The cuticle is formed by the secretion of chitin, which is an organic polymer that then organizes itself into fibrils that are subsequently sheathed by protein complexes binding to the chitin. Because the fibrils can slide alongside each other, a chitinous cuticle would still be quite flexible. Sclerotization is a process where enzymes create crosslinks between the protein sheaths that stops the fibers from sliding and thus creating a crystalline lattice. To retain flexibility, this hardening follows an oscillating pattern in order to create external segmentation with narrow flexible bands interrupting the sclerotized areas. Whenever an arthropod molds, this process is repeated because the newly formed cuticle starts out soft and flexible and will only harden once the old cuticle is shed. In many later arthropods, the sclerotized chitin was additionally fortified with minerals such as calcium carbonate, for instance in decapod crustaceans, such as crabs. It seems plausible that cuticles dominated by chitin or a similar substance are more susceptible to arthrodization than other kinds of cuticles. Annelids are in many ways similar to arthropods, but their cuticles are composed primarily of collagen instead of chitin. Chitin is a polysaccharide, much like the cellulose used by plants, and its linear chains can form strong hydrogen bonds and interact with proteins to create a rigid structure. Collagen, on the other hand, is a protein that forms triple helices and is therefore inherently more flexible and less prone to hardening itself. Any hard parts known from annelids, such as the jaws of some polychaetes like sand strikers, are achieved by other kinds of proteins such as glycine and histidine. These proteins can form strong crosslinks and even incorporate metal ions into their structure. Now, acorn worms are also burrowers like annelids and priapulids, but rather than relying on a tough cuticle based on either collagen or chitin, their epidermis is protected by a thick slime. Made chiefly of mucopolysaccharides, this slimy layer protects the skin from abrasions while at the same time being co-opted for carrying food to the mouth. This is particularly interesting because acorn worms are models for ancestral deuterostomes, marking the lineage towards both vertebrates and echinoderms, amongst other groups. Rather than investing in external toughness, like in the main protostome lineages, one could argue that deuterostomes relied more on a slimy outer surface and structural support from within. The bodies of acorn worms are strutted up by several internal structures, most notably a wishbone-like element supporting the proboscis and cartilaginous arches around the gill slits. However, they have something else very interesting: tiny dermal bones called ossicles. These are small nodules of calcite right below the skin surface. Their function is unknown, but often presumed to be anti-predatory or help providing grip during burrowing. There are genetic markers that indicate that these are homologous to those found in the closely related echinoderms. Both kinds of ossicles are excreted by sclerocytes and usually made of calcite, which is a special form of calcium carbonate crystals. But in echinoderms, the ossicles can grow to great sizes, often forming a subdermal skeleton of intercalated plates and jointed segments of enormous variety. The calcite of these ossicles is deposited as a specialized mineral form called sterium, which is highly porous and dynamic with active sclerocytes allowing for repair and further growth. Echinoderms represent the earliest known forms of biomineralization among deuterostomes, although as indicated by acorn worms, it may already have started earlier. The so-called small shelly fauna are assemblages of hardened elements, usually under 2 mm in size, and mark the transition to the Cambrian from the preceding Ediacaran period. Although tentative attempts at classification have been made, the identities of most remain elusive. Still, these may indicate that, in bilaterians at least, biomineralized skeletons may have started out as small elements distributed all over the body. We already discussed how the tiny ossicles of early deuterostomes grew into the large jointed elements of echinoderms that in some cases completely boxed the body into a rigid structure. Something similar seemed to have happened in other lineages, such as the one leading up to molluscs. Early Cambrian creatures such as Wiwaxia bore a chainmail of densely spaced, flattened spikes on their backs as they crawled over substrates. And despite not being molluscs, the bivalved brachiopods, nowadays sessile, seem to hail from similar slug-like creatures called tomatiids, adorned by many different mineralized, spiky elements. Even vertebrates went through a similar development, with small dental scales on the skin eventually differentiating into external plating and oral teeth respectively. The external biomineralized armors of early fishes eventually gave way to sturdy internal skeletons made of either pure cartilage or almost fully ossified into bones. So what overarching themes and patterns can we derive here, which we could transfer to evolution of life on other planets? The pathway to skeletons for all Earth-like life elsewhere may follow very similar principles. There are the following aspects we want to take into mind here: initiation, zoning, patterning, and growth. First, initiation. or the question of what might have induced the incipient stages of its evolution. Skeletons would start microscopically with the production of long organic macromolecules. The more macromolecules can be produced and bundled, the more firmness can be created. Hardening could be achieved by sclerotization or biomineralization, or a combination of both. Biomineralization may have started out in evolution as concretions of metabolic waste, so a byproduct that was then eventually repurposed. The selective pressures involved here probably center around predation, because even a slightly more crunchy animal would be less palatable, and so it could take off from there. Apart from providing protection, skeletons also provide better leverage for musculature, easily allowing for more powerful and faster movements, as well as larger bodies. Now, zoning involves the targeted formation of skeletal structures in distinct regions of the body. This could be externally, outside the skin, like a cuticle or shell, or internally as endoskeleton. Lifestyle is greatly influential here, and evolutionary shifts in lifestyle make for interesting outcomes. Slug-like substrate grazers would want to concentrate their protection dorsally while keeping the bottom side relatively soft and used for locomotion, like snails still do today. When the ancestors of cephalopods then took to swimming, this pattern persisted, leading to their unique body plans and eventually then losing their shells completely. Burrowing animals, however, would want a tough exterior all around to protect their bodies. Hardening of this exterior would then give rise to exoskeletal body plans like that of arthropods. Zoning is also involved in a gradual skeletonization of the entire body, one region at a time, over the course of evolution, as we see in the lineage leading to arthropods especially. For actively moving animals, hardening skeletons would have to retain some flexibility, so patterning is important here. In an earlier video I discussed how interfering gradients of morphogens induce regular spacing of elements, and this would have been helpful in the even distribution of skeletal elements. Finally, as smaller skeletal elements consolidated into bigger ones, or more flexible skeletons became more rigid, mechanisms need to be in place to allow for growth. Arthropods inherited the molting strategy, replacing their exoskeleton every time they outgrow it. Molluscs solve this issue by simply extending their growing body outwards, excreting new materials on the edges of an ever-widening shell to accommodate this. Endoskeletons typically need to be made of more dynamic materials, being constantly rebuilt, as is the case in echinoderms and vertebrates. Interestingly, the skeletons of echinoderms, though principally endoskeletal, are so close to the surface that they often functionally act like exoskeletons. However, because it's embedded and open to reworking by the sclerocytes, it's much more adaptable and does not constrain the body like the arthropod exoskeleton does. Now, skeletons clearly define an animal's body plan at a very fundamental level, and subsequently this would lead to its relative success in the niche of the biosphere it will develop in. Given all of the above, it would seem that on other planets very similar to Earth, you would likewise end up with creatures very similar to annelids, molluscs, arthropods, and perhaps even vertebrates. And just as much as size matters, gravity matters. Under Earth-like gravity, there are major size constraints to exoskeletons, especially on land. So any arthropod-like creature championing exoskeletons would remain small, while endoskeletal land creatures would be able to grow to much greater sizes. Exoskeletal animals could still grow into impressive sizes on low-gravity planets. But if we ever were to visit them, we would have superpowers compared to them, somewhat like how John Carter would be a superhero on his fictional Mars. What is also important is geochemistry. Biomineralization would only be possible in planetary environments with exposed land masses subject to weathering to maintain steady concentrations of minerals like silica and calcium carbonates. So on ocean planets, if life is even possible there, organisms will have to conserve their precious minerals and opt for fully organic skeletons instead. Yet, even with biomineralization being very limited on those worlds, creatures like octopuses are evidence that hard skeletons are not strictly required for evolving large and highly complex life forms. But what if a developing biosphere would find itself in the opposite situation, and with carbon and perhaps even water levels dwindling, it would have to rely increasingly on minerals instead? That will be the topic of an upcoming video on crystalline alien life forms. And what do you think? Would the basic laws of physics always render evolving creatures in the same skeletal categories as we see on Earth? Could we conceive of alternate biospheres with different structural requirements? Let us know in the comments and I will take a look at it. If you like this video, please give it a thumbs up and maybe even a sub if you haven't already. Thanks for sticking around. Until next time, stay sharp and stay evolved.